Hi everyone, I'm Stephanie. We're a team that review the darker secrets from like the sugar galaxy exploration. So our project focuses on follicular lymphoma, which is like a very slow-growing cancer that starts in the follicles and like the lymph nodes. So it is like the place where our body reacts and fights for against infection. So the lymph node follicles are clusters of like white blood cells or B cells that are located in each of these lymph nodes. So on the right, it is showing the lymphatic systems of the body and also showing where the lymph nodes are located. So basically, it means that like follicular lymphoma could happen in any of these places. So it's like affecting the entire body. So treatment is available for follicular lymphoma. However, some of the patients like experience like relapse or like they're non-responsive to the treatment. And even worse, some of them will develop like into a more aggressive like lymphoma, leading to a poorer prognosis. And there have been like multiple studies on follicular lymphoma, but then one of which is kind of like identifying an unusual glycan, which is like a sugar complex structure on like the B cell surface. So glycans are complex sugar molecules that is attached on the proteins on the cell surface. So it's kind of like these kind of like hairy looking structure on the diagram on the right. And so they help the cell communication and also regulate immune responses. So in follicular lymphoma, the abnormal glycans that was found is actually supported the continuous activation signal that then leads to uncontrolled proliferation of these B cells and then contributing to follicular lymphoma. So in this project, we aim to visualize these sugar directly on the tissue samples from the patient and then identify how the change in the patterns and also understand how these would affect immune responses and also the development of follicular lymphoma. So we're a team that combines like people from labs at MIT, BIDMC, and URMC, and then we together develop a novel imaging platform that can detect both glycans and proteins at the same time, that we name it as glycoscope. So we started with like a tissue that is like directly extracted from the patient. And then we use these kind of like oligos, which are like DNA fragments, to barcode antibody, which are like protein sensors, and then the lectin, which are like the glycan sensor. And then we stain it on the tissue. And then we put it onto the imaging machine, and then with like iterative cycles of like imaging, we'll be ending with like 50 to 60 proteins and glycans targets that is present on these tissue. So this is like the image that we got from using glycoscope. And you can see that these are, each of the circles are showing the follicles from patients that is like non-cancerous, like they're healthy, compared to like follicular lymphoma patients. So you can already see like very drastic differences on how they look on this image. And when we try to zoom in, so you can see that like the healthy follicles, the green are actually showing the sugar that is of interest. It is like showing a very like a round, like an organized structure appearing as like a ring-like structure that is like surrounding the red, which is like the follicle center. But then when we look at like the follicular lymphoma, it's like everything changes. So the organized structure is being like disrupted, and then like the green is kind of like going into the red. So you can see a mix of like green and red together. So it's kind of like a very distinct disease-specific glycans redistribution. And later on, we found that this redistribution is actually important to suppress immune responses and leading to a more pro-cancer microenvironment in follicular lymphoma. And then we're now continuing with this disease, with this project, and then applying glycoscope into more of like the patient samples, and then trying to link whether this redistribution of glycans is linked to the severity of like the disease, and then which we can help with like patient stratification and also the early detection of this cancer. So with that, I would like to thank all our funding sources, like really supporting us to push this forward, like push this project forward. And yeah, thank you so much for coming in tonight. And yeah, we're the Sugar Galaxy like exploration team. Thank you.